For the next presentation is Tannic acid attenuate nanoplastic-induced neurotoxicity in silico and in vitro integrate study by Nonthaphon Hiran from Faculty of Medicine, Princess Naradhiwas University.
Good morning, fellow participants and respected judge. My name is Nonthaphon Hiran from Princess of Naradhiwas University. I'm here to present to you about tannic acid attenuate nanoplastic-induced neurotoxicity in silico and in vitro integrated study. Plastic pollution is a pervasive problem, extending globally from large debris to microplastic fragments, such as microbeads found in cosmetics or as a result of breakdown from plastic bottle or clothing. Data from 2019 show that globally we generate hundreds of millions of tons of plastic waste, including 15 million tons of polystyrene that are usually harder to recycle and remain in our ecosystem. But what happens when this tiny particle reaches? Nanoplastics, in this case polystyrene nanoplastic, can enter and accumulate in vital organs and cross into the central nervous system, where they can interact with various neuronal cells. This interaction triggers cellular stress, characterized by mitochondrial dysfunction. This results in oxidative state through generation of reactive oxygen species or ROS, which cause oxidative damage, stimulate the release of pro-inflammatory cytokines such as interleukin-6 and TNF-alpha. This cascading event causes cascade activation, resulting in apoptosis, ultimately leading to neurotoxicity and neuronal damage. Facing this widespread threat, we sought a readily viable countermeasure. Our attention turned to tannic acid. Tannic acid is a natural plant-derived polyphenol known for several beneficial biological activities. It has established antibacterial and wound healing properties. It possesses antioxidant activity capable of reducing ROS, and also exhibits a potent anti-inflammatory effect by inhibiting pro-inflammatory cytokine. Given its ability to target the core mechanism of nanoplastic toxicity, tannic acid became our candidate of interest. The objective of our study is to investigate the potential neurotoxic positive effect of tannic acid against toxicity caused by amine-modified polystyrene nanoplastic in human neuroblastoma cells. To achieve this, we employ an integrated study design. In vitro cell experiment, with in silico computational modeling. Our in vitro model utilized neuroblastoma cells, treating them with aminated polystyrene nanoplastic alone or in combination with tannic acid. We assess cell viability using MTT and LDH assay, cellular uptake and apoptosis using flow cytometry, and finally inflammatory response by measuring interleukin-6 level via ELISA. In parallel to this, our in silico approach utilized network pharmacology and molecular docking to predict the underlying molecular mechanism of tannic acid interaction with key cellular target. First, we established the cellular toxicity induced by the nanoplastic. We found that aminated polystyrene nanoplastic induced concentration-dependent toxicity in the neuroblastoma cell with an IC50 of 52.08 microgram per milliliter. At a concentration of 50 microgram per milliliter, the cellular viability and morphology was significantly compromised. Furthermore, exposure led to a substantial cellular uptake of the nanoparticle, promoting apoptosis, and resulted in significantly increased inflammatory cytokine interleukin-6 compared to the control group. This experiment showed that nanoplastic can cause a detrimental effect on the health of neuronal cells. After that, we confirmed the safety of our proposed treatment. We found that tannic acid at a concentration up to 20 micromolar exhibits no cellular toxicity, as confirmed by MTT and LDH assay. Based on this result, a concentration of 20 micromolar for tannic acid and 50 microgram per milliliter for nanoplastic were the selected dosage for our combination experiment. With the inflammatory response caused by internalization of polystyrene nanoplastic, what occurs when aminated polystyrene is treated in combination with tannic acid? Our findings show that tannic acid can protect the cell. When the cell was treated in combination with our selected dosage, cell viability was significantly improved compared to nanoplastic only group. In addition to the reduction of aldehyde release, tannic acid also significantly reduced the cellular internalization of the nanoplastic. Furthermore, tannic acid significantly reversed the induction of apoptosis caused by nanoplastic. Regarding the inflammation component, tannic acid treatment also significantly reduced the elevated level of interleukin-6, bringing them back toward the control baseline. Next is our in silico experiment to provide insight into how tannic acid achieves its protective effect. Our network pharmacology analysis identified 20 common targets linking tannic acid and polystyrene toxicity. These targets implicated pathways associated with oxidative stress and antioxidant defense. These targets were then analyzed using co-enrichment analysis and were found to be connected to the response to ROS, a response that leads to cellular inflammation and apoptosis. Next, we used molecular docking to screen our target protein in oxidative stress pathway and found that tannic acid has strong binding energy with KEAP1, with an energy level comparable to that of a known KEAP1 inhibitor. This finding strongly suggests that tannic acid can achieve the pathway by binding to KEAP1 and boost the cell antioxidant defense. This may be one of the pathways which tannic acid can help reduce the inflammation of the renal cell. So we concluded that tannic acid exhibits neuroprotective effect against ME-modified polystyrene nanoplastic-induced toxicity by inhibiting nanoparticle uptake, reducing apoptosis, and possibly activating the antioxidant defense well enough to keep one pathway. This finding highlights tannic acid as a promising candidate for protecting against polystyrene nanoplastic-induced neurotoxicity. The significance of our study is that we show that natural compound can protect the human brain cell. We identify tannic acid as a safe pan-derived neuroprotective agent. We provide a proof concept to reduce the neurological risk of plastic pollution and lower typic insight that can inform public health and environmental policies on the danger of plastic waste. Although our study has some limitations, such as only being an in vitro study and does not fully replicate the physical challenge of the human body, and only use a single type of nanoplastic, which is not a true representation of the reality of plastic pollution, future mechanistic studies are still warranted. We should test tannic acid efficiency against a wider variety of nanoplastic for more relevant results to river pollution. And additionally, in vivo study can provide a deeper mechanistic insight to confirm tannic acid as a cost-effective agent against plastic pollution. Thank you for your attention. I would like to, any question will be appreciated.
Can you show the any results again how tannic acid prevent toxicity induced by nanoplastic in your hypothesis or your proposed result?
This one? Right.
Tannic acid, okay, here. Cell viabilities, and then you present the effect intracellular, right? That is interact with different pathway. But my concern is if you show the structures of tannic acid in some slide. Yes, here and also others, the previous one as well. It's quite large, and how this molecule go inside the cell. Do you have an idea or the possibilities that tannic acid can go inside the cell and interact with your target in reality?
In reality, tannic acid there is a research that shows tannic acid can absorb to the gastrointestinal system and can go into the renal cell, but the crossing into the blood-brain barrier needs to be further studied.
But is there an evidence that it is exists as an original form in the plasma?
Plasma.
Yes. So if you can go back to the structures in previous slide, whatever. Yes. Here, yeah. I'm not so sure whether intestine can absorb it originally, and these whole structures can present safely, in the plasma. But anyway, I don't know. But my suppose is that maybe it's cleaved, and some of this branch is real effective molecules.
Yep.
So at your molecular docking slide, how could you select the target of the, I'm not sure, is the binding site or the target protein? So how do you select this?
The binding site.
Molecules?
I use the website to show the area of binding energy, which is the most promising to the binding area, and I try to use a different site. Various sites, I use about 5 sites to test the binding energy of the tannic acid and repeat it about 3 times. Yeah.
So you conclude that KEAP1 is the most selective binding, right?
Yeah, I find I use various proteins from this one and find that KEAP1 have the best binding energy.
Okay, so how could you validate this data in the future?
We can
Yeah.
further validate this study by testing for the antioxidant activity of the cells or by silencing the key one pathway and testing for oxidative response in the treatment of the cell in tannic acid to see that tannic acid can really help reduce the oxidative pathway or using other pathway or using protein from other pathway that related to tannic acid. Thank you.
Thank you for your excellent presentation.